So the endoplasmic reticulum is a very special organelle in the fact that it is going to direct materials where they need to go. So the endoplasmic reticulum actually is going to attach to the outside of the nucleus. And the reason for that is the nucleus is going to be a place where a lot of materials are produced. So within the core of that nucleus, you have proteins, you have RNA, you have other things that are being made. And once they're made, they need to be shipped to their final destination. And that's what the ER is going to do. So again, if you are kind of visual, if you think about our classroom at St. Luke's. So when you guys come into the building, in order to get to our classroom, you have to navigate a series of hallways that are going to eventually deliver you to our classroom. So if you kind of think of the classroom as being the nucleus and the hallways as being the endoplasmic reticulum, that is the exact same design within the cell. So you have the production of materials happening in the nucleus. Once they're made, they're shipped out of the nucleus and they will be directed into the right hallway, so to speak, the right endoplasmic reticulum. And the endoplasmic reticulum will then navigate through the cell and deliver that product to where it needs to go. It might be another organelle somewhere in the cell. It might need to be shipped out to another place in the body. So the endoplasmic reticulum might lead it out to the cell membrane and ship it out of the cell completely. But the endoplasmic reticulum is essentially a network of hallways built within the cell so that products are going to be delivered to the right destination. From there, we have the nuclear envelope. The nuclear envelope is very similar to the cell membrane. It's just wrapped around the nucleus. So we already talked about the nucleus. It's the core of, or at the core of the cell, and it's where a lot of materials are produced. The nuclear envelope is going to have tons of pores, all these openings, so that as those products are produced, they have a place where they can exit the nucleus and get to where they need to go. Now, one of the most important things that is held in the nucleus is your DNA. And the DNA is actually so large in size that these pores, even though they're all over the nuclear envelope, the pores are so small and the DNA is so big, the DNA can't get out. So we always keep the DNA housed in the nucleus just for protection. From there, we have the vacuoles. Vacuoles are basically storage tanks that you guys have within the cell. And it doesn't really sound that exciting that you have these vacuoles that are going to hold water, but they actually serve a very important purpose. There are tons of chemical reactions that happen within the cell, and those chemical reactions have to happen within the right environment. If you had a lot of excess water floating around in the cell, it would be the equivalent if you made Kool-Aid and put in twice as much water as you were supposed to. You guys know that it would dilute it down and the taste would be very flat. So likewise, within the cell, if we're trying to get these chemical reactions to happen, they need the right environment, they need the right conditions and concentrations. And so that water can't just be allowed to go wherever it wants to because it would dilute the cell to the point that those chemical reactions would never happen the way that they need to. So these vacuoles are very important to hold any excess water that is in the cell to maintain the proper conditions so that chemical reactions can happen the way that they need to. If you move on to the second slide that talks about organelles, the next one is vesicles. Vesicles are going to be another area within the cell where we can hold liquid. This tends to be liquid that is not water. So anything else that would be in liquid form that kind of needs to be contained would be held within those vesicles. And these can even include neurotransmitters. So hormones or chemicals that your body needs to release at very specific times would be held within these vesicles. And when we get into our nervous system chapter, we will talk about those vesicles a little bit more. So it's not necessarily, it's like a waste product fluid that it's holding, but it can be holding a chemical or a hormone from the body that needs to be maintained until you actually need that hormone to be released. The next one we have are microfilaments. Microfilaments are protein-based. Again, we see protein. It's a common theme in a lot of the things that are made within our body. but these protein-like rods, which are going to serve or be the equivalent of your skeleton. So we have our skeleton, which gives us the framework and the shape of our body. And likewise, to help maintain that circular shape that we think of a cell having, you do have these microfilaments. And what they actually do is they'll create a framework around the outer edge of the cell to help support it and keep that circular shape. Now, the one unique thing about the microfilaments is that they are not secure in one particular location. So if the cell needs to restructure or reposition itself, it can actually dismantle these microfilaments, break it down, and move them to a different location, and then rebuild it as needed. And that's obviously not something we can do with our skeleton, but the cell has a little bit more flexibility in what it is able to do with those microfilaments. From there, we have the flagella and the cilia. These are two extensions that you have off of the surface of the cell, and they are a little bit different. And the reason we give them different names is because they serve different purposes. Flagella, you typically have one if you have it on a cell, and it is going to be primarily for movement. So it's a very long extension that you have off the surface of the cell, singular if you have one present, and is going to whip back and forth and provide movement or locomotion for the cell. So it's trying to get it from one location to another. Cilia are a little bit different. They are shorter in nature. You typically have tons of them on the surface of the cell, and they are going to be used to clean debris so that it doesn't build up around the exterior of the cell. So they will again kind of whip back and forth, but it's basically like a broom. It's constantly moving debris and keeping it from being able to build up and create a toxic environment around the cell. Then we have the Golgi apparatus. Golgi apparatus is usually defined in a textbook as a stack of flattened membranes, which is a little bit hard to kind of wrap your mind around. What the Golgi apparatus is actually going to do is it is going to be like the gift wrapper of the cell. So we've talked about all these products that the cell is producing. And once the cell has produced them, they need to get shipped out to wherever they're going to go or wherever they're going to be used in the cell or in the body. But the problem is that a lot of times they have to have these finishing touches put on them before they leave the cell or before they reach their final destination. If you buy somebody a birthday gift, you don't just buy it, take it off the shelf, and hand it to them. What do? You wrap it up, you put a bow on it, you might attach a card to it. So you put all these finishing touches on it before you actually deliver the gift to the person. That is what the Golgi apparatus is going to do. So the cell has produced a protein, and that protein needs to be delivered somewhere else in the body. What the Golgi apparatus will do is it will typically attach a protein, a carbohydrate, or a lipid to the product. If it is going to be traveling a long distance, it might attach carbohydrates so that it has an energy source in the travels to get it to where it needs to go. It might take a protein as an additional building block to be used once it gets to its final destination. It can take a lipid for protection. So whatever it needs in order to safely get it to its destination within the body or the cell, that's what the Golgi apparatus will attach to it. Then we have the mitochondria. For some reason, the mitochondria seems to be one that people just remember. If they've ever had a background or a class where they've dealt with organelles before, they seem to remember this one. The mitochondria is the powerhouse of the cell. This is where energy production is going to take place. Now, ATP is our big fuel source in the human body. It is going to be what gives your cells the energy they need to do all this work that we've been talking about. And so the mitochondria is going to be the actual location of where these ATP molecules are created or produced. And then we have these centrosomes. Centrosomes are two structures. They typically are found floating around the outside of the nucleus. And there's not a whole lot really known about them. They're still doing a lot of research on everything that the centrosomes do. But a lot of times they are just floating around the outside of the nucleus. The only time they move from that position is during the process of cell division. And in the process of cell division, one of those centrosomes will go to the North Pole, so to speak. If you think of this nice round cell, the equivalent of our Earth, we have a North Pole and a South Pole. Same thing with your cells. You have a North Pole and a South Pole. So one of the centrosomes will go to the North Pole, the other centrosome will go to the South Pole, and they will aid and assist in moving things in the cell for the process of cell division. And then the last one we have are lysosomes. So lysosomes are the garbage cans of the cell. Your lysosomes are going to be filled with a very strong digestive enzyme. And when something dies or becomes damaged within the cell, it is going to get put into this lysosome, coated in this digestive enzyme, and it will eat it and break it down so that you don't have this garbage building up in the cell. So like I said, if an organelle becomes damaged or if it dies just from wear and tear, what will happen is it gets transferred into this lysosome, coated in that digestive enzyme, so it deteriorates and you don't get all this clutter building up. So I know that there's a little bit more with this packet, but we are going to pause our conversation right there. And on the test, you are only going to be responsible for the material through the organelles. So you have your intro packet, you have your chem packet, you have the tissues, you have metabolism, and just the first basically page and a half of this packet on cells. Keep your cells packet because we will pick this up after that test and we will continue on with the rest of the information here, but you will not be responsible for anything after that second organelle slide from your cells packet. Please make sure that you bring all those with you to our next face-to-face meeting. If you have any questions between now and then, please just send me an email and I will get back to you. If you don't hear from me in 24 hours, please try again because I did not get your email if it's taken me longer than that to respond to you.